Hi, I'm Daniel Kang, and we're in Dr. Thomas White's CS201 Intro to Technology, Section 26 class. These are my lovely partners.
I'm Chandler Norris.
I'm Jessica Street.
And we're excited to present you my research, which is on expression of heat shock protein 70 in the zebrafish Danio rerio in the state of Alabama. And so we'll begin. A brief introduction. The zebrafish is just a small organism that's found in parts across the world, specifically originated in parts of India. And it's just the best specimen to do research on for heat shock protein 70. Please take it away.
Okay, so for our introduction, this is a relatively understudied research topic. Several organisms possess forms of heat shock proteins. HSP70 is the focus of this research. The background: HSP70 acts as a chaperone to assist with repair of damaged proteins. It also aids in regulation. This HSP is influenced by abiotic and biotic factors. So our research specimen, like Daniel said, is the zebrafish. It is called the Danio rerio. It originates in the Ganges River of India and other freshwater streams in surrounding countries. It's an invasive species in Alabama. It has a fast generation time. It takes only about 3 months for this species to mature. And it's small in size and can withstand a wide range of temperatures. So what are some medical applications for HSP70? Further research of HSP70 creates a broader field for tissue regeneration techniques in medicine. So obviously tissue regeneration, if you get a burn and we're trying to regenerate that tissue that got burned off, it can be used for that. Manipulation of the HSP70 allows more treatment options for diseases such as cancer and other tissue-damaging diseases.
Now we get into the fun part of the research, some tools and methods. So some very specific methods go into this project. So one of the first things you need to do with the fish is you need to take tissue samples. And we want to take tissue samples from the skeletal white muscles and the gill muscles of the fish, because those provide the best samples. So we'll take those tissues and we'll homogenize them. So we'll basically crush them up and we'll get really homogenized tissue. And we will have 3 different samples. We'll have a cold sample, a room temperature sample, and a hot sample. So they'll be at 3 different temperatures to observe the effects or the expression of the heat shock protein 70. So after homogenization, one important thing is to centrifuge the tissue samples. That means we'll have to rapidly spin the samples around, basically, so it kind of separates the different tissue layers. So we're trying to get to the proteins. So we'll do that. The next thing we'll do is we'll do 2 different types of SDS pages, which is just running this particular protein sample on a gel and separating it through different dimensions, one by its weight and one by its different chemical and physical properties, so that we can find the specific heat shock protein 70 on the samples. Finally, to completely isolate the samples and to actually observe where the sample is, we'll do a Western blot, which is basically stating the protein samples which are on the gel, and taking that off, and we'll have a sort of something to observe and see where the actual protein is. So this is an example of the results of one such experiment. So as you can see, this bar right here, the subtle different, the dots that form, that's actually the heat shock protein 70. This is an example of a Western blot. That's very interesting. So how they are determined, like I said earlier, physical and chemical properties. One physical property of heat shock proteins, they're measured by their weight. So They measured in kilodaltons, so 70 refers to 70 kilodaltons of the protein. So after the samples are taken and observed, the way you can determine which one is heat shock protein 70 is just on its place on the blotting gel or the electrophoresis gel, so you can determine where it actually is. And that's about it. And Jessica here will talk about the budget for the experiment.
Okay, I'm going to talk about our budget. The proposed budget is $2,588. The equipment we need is a tetra blotting module, electrophoresis and blotting apparatus, biorad, and it costs $630. The various chemicals are the Fisher Scientific, Sigma, Altridge, and biorad, and it costs $1,958. And now I'm going to talk about our acknowledgments. So we want to thank Jacksonville State University for providing our facilities and laboratory and computers. We want to thank Dr. Mark Mead and Raheem Zatili for providing laboratory procedures and background on the zebrafish.
Now this is a very important topic to me, and I believe that with further research, since this is not a widely known topic, as of now, more research needs to be done, and better medical applications, and just to the betterment of mankind and to the posterity of our health and everything. This can do a lot to change the game of the medical field. So please consider this when you are thinking about things to donate. And I need money for this, so thank you for your time, and we appreciate your patience.